So what do you mean by immunity? Immunity is a Latin, it has been derived from a Latin word called as immunitas. The meaning of this is freedom from disease. Okay. So it is defined as resistance offered by the host against any foreign antigen or any foreign substances. Okay. So immunity means it is the resistance offered by the host against a foreign substance like any of the infections or antigens. The resistance what we offer against a foreign substance, it can be present right from the birth, or it can be developed, it can be acquired during the course of the life. I'll ask you one question. If a newborn baby is been exposed to some infection, can it develop any kind of immunity to the infection? Yes, that is called as innate immunity. Okay, so immunity can be of two types, innate immunity and acquired immunity. Innate immunity means the resistance offered by the host against the foreign antigen, which is present right from the birth. Whereas acquired immunity is the resistance what we develop during the course of the life. Okay, we'll discuss more about this in the subsequent slides. So first we will focus on innate immunity. Okay, so as I have told you, innate immunity is nothing but it is a resistance developed by the host towards an infection which is present right from the birth. And this is usually due to the genetic or constitutional makeup of the individual. Genetic or constitutional makeup of the individual. Okay. Innate immunity usually occurs in minute. Okay. When a newborn baby is exposed to some infection, he can develop some kind of resistance towards the infection. This is called as innate immunity. For example, the classical example of innate immunity is, the classical component of innate immunity is phagocytes like neutrophils. So when an infection occurs to a newborn baby, the neutrophils first will migrate to the site of action. So this migration takes place in minute. That is, innate immunity occurs very soon following the antigenic challenge, and there is no prior exposure to the antigen is required. Even for the first time when the antigen or when the infection occurs to the newborn baby, he can develop innate immunity and the phagocytes can go to the site of infection in minutes. Prior exposure to antigen is not required. Okay. Innate immunity is non-specific. Very, very important. The most important difference between innate immunity and acquired immunity is acquired immunity is specific. The components of acquired immunity are T cells and B cells. These are very, very specific. Each T cell and each B cell are educated to develop immune response towards a particular antigen. In contrast, the components of innate immunity are non-specific. Example, phagocytes. The same neutrophil can go to the site of infection by any of the organisms. Okay, or whatever organisms can infect, the neutrophils can go to the site of infection and they can try to provide some sort of protection. So they are non-specific. They can be stimulated against any diverse group of infections. And though they are non-specific, the diversity of innate immunity is limited. In contrast, acquired immunity, the diversity is wide. In contrast, the innate immunity, the diversity is restricted. The cells mediating innate immunity like the phagocytes or NK cells, they can act only against the few limited type of antigens, epitopes. And one more important difference is there is no immunological memory in innate immunity. Innate immunity doesn't have any immunological memory. The meaning of which is, if the same infection occurs again and again, then the innate immunity response will remain the same. It will not be faster in the subsequent infection. In contrast, the acquired immunity, the immunological memory will be there following the primary contact, so that if the same infection occurs again and again, then the response, the acquired immunity response will be faster in the subsequent contact. So what are the various microbial antigens, and they react to which host cell receptors in innate immunity? The classical example of microbial antigen which reacts with the host receptor is MAMP antigen, which is nothing but it is microbe associated molecular pattern. This is the antigenic unit of the microorganism. They are the antigenic unit of the microorganism which will bind to the corresponding receptors present on the host cell to initiate the immune response. So the host cell receptors which can bind to the MAMP antigens, they are, example of those are called as pattern recognition receptors, the classical example of this is toll-like receptor. Toll-like receptor. We will discuss about this in the subsequent slide. So these are the basic properties of innate immune system. So now we will discuss how the mechanism of innate immunity works. So the first important thing is receptor interaction. As I told you in the previous slide, there is a particular microbial antigen. Okay, so if any infection, any microorganism infects into the body, it will initiate immune response. Innate immunity is the first thing to develop. Suppose for example any of the component of innate immunity like phagocyte will be coming to the site of action. So when the phagocyte will come to the site of infection, what happens is the microorganism will have certain antigens on their surface. These are antigens which can mediate innate immunity. Those antigens I have told you in the previous slide, they are called as MAMP antigen or micro associated molecular pattern antigens. These molecules, they bind to the corresponding host cell receptors. The corresponding host cell receptors, host cell receptor means the receptors which are present on the host cells which will mediate innate immunity, like for example phagocytes or NK cells. Those receptors are called as, I told you, they are pattern recognition receptors. Pattern recognition receptors are nothing but the classical example is toll-like receptor. Toll-like receptor. And the classical example of MAMP antigen, that is the microbe associated molecular pattern antigens, the classical examples are the peptidoglycan, lipopolysaccharide, teichoic acid. Okay, you know that all these are present on the bacterial surface. So all this lipoprotein, all these are the classical example of microbe associated molecular pattern, MAMP antigens. So these are the antigens of the microorganisms which can initiate innate immunity by binding to the specific receptors on the host cell which are called as pattern recognition receptor. The classical example is toll-like receptors. So this is the first step of innate immunity. The first step of innate immunity, what we have discussed in the previous slide, that is nothing but the microbial surface molecules like the bacterial LPS, flagella, or maybe any of the other structures. They bind to the pattern recognition receptors of the host cell. They are nothing but the toll-like receptor. What happens after the binding? That we'll discuss now. What happens after the binding? When the receptor interaction take place, it activates various transcription factors. So please remember the following binding of toll-like receptor to MAMP molecules. What happens is there is a generation of signal. Okay, signal gets generated, and this signal is responsible for activation of various transcription factors. And these transcription factors, they in turn stimulate various genes responsible for encoding several cytokines and enzymes, which in turn are involved in various antimicrobial activity. They are responsible for destruction of the microorganism. Okay, so how innate immunity take place? Step 1 is when a foreign infection occurs, the components, the cells mediating innate immunity, they go to the site of the action, like for example NK cell or the phagocytes. When they go to the site of the action, the first interaction is the receptor interaction. The toll-like receptor of the host cell binds to MAMP molecule of the microbial surface, following which the signal gets generated. The signal in turn activates transcription factors. Now we'll discuss various components of innate immunity. You have a question for you. Which is not a mediator of innate immunity? Based upon what we have discussed so far, which of this following is a mediator of adaptive or acquired immunity? Which is specific here? Yes, you are right. The specific here is T cells. So T cells and B cells, they are part of acquired immunity. Whereas NK cell, neutrophil, skin and mucosal surfaces, they are non-specific. They are part of innate immunity. So let us now discuss the components of innate immunity. There are several components enlisted here. We will discuss one by one. The first to start with anatomical barrier such as skin and mucosal barrier. So what is the role of skin? You all know skin acts as a mechanical barrier. It gives mechanical protection against the organism to entry. Skin can have various antimicrobial effect, like you know that skin produces sebum, and sebum is responsible for various antimicrobial activity. It contains various antimicrobial peptides which can destroy the microorganism. And skin also has the ability of killing the microorganism by having intraepithelial lymphocyte, IEL, intraepithelial lymphocytes, which are present on the cutaneous surface. They can destroy the microorganism. Okay, so these are about the skin properties. Coming to mucosal membrane. Mucosal membrane can produce mucus. Mucus has a property of entrapping the microorganism. It can entrap the microorganism. It can trap the microorganism. And other anatomical barrier like cilia. Cilia is usually there in lower respiratory tract surfaces, and they have a property of propelling the microorganism outside. They remove the microorganism outside the respiratory tract. Okay. Normal flora, the normal bacterial flora, the normal microbial flora, they are present in the surface of gastrointestinal tract and also respiratory tract. They are responsible for various activities, like they compete with the pathogen. They compete with the pathogen to enter, and which is responsible for limiting the invading microorganism, because the normal flora is always, it helps in blocking the microbial entry. Okay, so these are the various anatomical barriers. Now we'll discuss about the physiological barriers. like the temperatures, low pH and various secretory products of the mucosa. So you know that the human body temperature is 37 degrees centigrade. This temperature inhibits microorganisms to grow, especially the thermophilic or the psychrophilic microorganism which cannot grow in the body. Those organisms can be inhibited by the body temperature. Low pH, for example, the gastric juice has a property of low pH, and this low pH can inhibit the microbial entry. Various secretory products of the mucosal surface, like just now we have discussed the gastric juice, which contain various enzymes like pepsin, trypsin. All these enzymes can destroy microbial peptides. Saliva also has some antimicrobial property. Tears also can have some antimicrobial property. So all these are example of innate immunity. They are non-specific. They can be produced against any invading microorganism. The next example of innate immunity include phagocytes. Classical example of innate immunity is phagocyte. They are nothing but neutrophils and macrophages. Okay, they mediate something called as phagocytosis. Whenever a microbial invasion will take place, microbial infection will take place, the phagocytes, they go to the local site. So the first step under phagocytosis is engulfment. What happens here is the microorganism gets engulfed by the phagocyte inside. Following engulfment, they will be trapped inside the phagocytes, inside the phagosomes. The phagosomes are present inside the phagocytes. Once the microorganisms enter into the phagocyte, they will be engulfed by the phagosomes, following which there occurs phagolysosome fusion. The lysosome gets fused with the phagosomes, so that the lysosomal content, they will be inside the phagosome, and that is responsible for microbial killing. The third step is microbial killing, mediated by various lysosomal enzymes. Okay, so this is how the phagocytosis takes place. The next important component is NK cell. NK cell are the component of innate immunity which is responsible for cell-mediated immune response. The cell-mediated immune response is usually mediated by the cytotoxic T cell. The cytotoxic T cell. However, if the antigen in a neonatal case where you need to provide neonatal immunity, innate immunity where there is no cell-mediated, the T cell-mediated immunity is available, in such case, the CMI is mediated by NK cells. The detail of NK cells we will discuss under cell-mediated immunity. Then comes to other rare classes of lymphocytes. You know that T cells and B cell are the classes of lymphocyte. They are the major classes of lymphocyte, which are the component of acquired immunity, as we have discussed earlier. However, there are certain rare classes of lymphocytes. For example, NK T cell. There are some T cells which are modified, which are called as NK T cell, and gamma delta T cell. Most of the T cell will have alpha beta receptor. Receptors. Whereas there are certain T cells which have gamma delta receptors. These are called as gamma delta T cells. They are usually present as intraepithelial lymphocytes. They are also part of innate immunity. Okay, they are not adaptive immunity, acquired immunity. They are part of innate immunity. Similarly, there are two variants of B cell. One is called as B1 cell, and the other one is called as marginal B cell, marginal zone B cell. So these two are the variant of B cells. Though they are B cells, but they are not a part of acquired immunity, they are part of innate immunity. So this is what you need to, they are rare classes of lymphocytes. Then the next example is mast cells. Mast cells is another example of innate immunity, what happens? Mast cells are usually present in respiratory mucosal sites. Okay, what happens is whenever any allergen enters, whenever any allergens enter into the respiratory mucosa, they activate the mast cells. Activation of the mast cell occurs by two ways. Mast cell on their surface, they may have toll-like receptors which can directly bind with the allergen. Step 1. Otherwise, mast cell may be coated with antibodies like IgE antibodies, and this allergen can come and bind to the IgE antibody, and through IgE mediated activation of the mast cell also may take place. When mast cell gets activated, they undergo degranulation. Degranulation of the mast cell leads to liberation of various cytokines, various secretory products which can destroy the invading allergen. This is how the mast cell works. This is responsible for a type of hypersensitivity reaction called as type 1 hypersensitivity reaction. Okay, so these are the components of innate immunity. There are few more components like dendritic cell and complement pathway. Dendritic cell, whenever a microbial entry will take place, dendritic cell respond to the microbial invasion and they produce various cytokines which can induce inflammatory response. Dendritic cell can also help in the transport of microbes from the site of entry, site of infection, and they take the microbes to the lymph nodes. Okay, so they can be responsible for transportation of the microbes from the site of infection to the lymph node. So this is also an example of innate immunity. Next is complement. Complement in general are part of innate immunity. They are non-specific. There are three types of complement pathways. The classical pathway, the alternate pathway, and the lectin pathway. Among the three pathways, alternate pathway and lectin pathway are example of the classical example of innate immunity. They are independent of antibodies, whereas classical pathway of complement, they are antibody dependent. So they can be considered as part of adaptive or acquired immunity, or in fact they act as a bridge. They act as a bridge between acquired immunity and innate immunity. Okay, so this is about complement. The detail of complement we will discuss later. They are the non-specific proteins present in the serum, and whenever any microbial invasion can take place, that can activate the complements. Complement activation leads to the destruction of the microorganism. Lysis of the microorganism which leads to microbial killing. Complements are in general, they are the component of innate immunity, except classical pathway of complement which act as a bridge between innate immunity and acquired immunity. The next component of innate immunity is inflammatory response, and here you know that whenever any infection takes place, for example here you have a tissue damage, you have a foreign body invasion, along with that some of the organisms are been invaded. So when any infection takes place, first thing what happens is the nearby vessels you will have vasodilation. The first step is you will have vasodilation, and after a vasodilation you will have leakage of the various plasma proteins. The plasma proteins will be leaked out, following which you will have recruitment of the phagocytes. The phagocytes, they extravasate. They will come out of the blood vessel and they will extravasate, following which they will migrate by the chemotactic activity and they will reach to the site of action. At the site they will do something called as engulfment. We have discussed in the previous slide What do you mean by engulfment? Engulfment has got 3 steps. Phagocytosis, that is engulfment of the microbes inside the phagocyte. Then it will go inside the phagosome. Then you will have phagolysosome fusion, following which microbial filling, that is destruction. Step number 5 is destruction. So this is how the inflammatory response will take place, and please remember the inflammatory response is nothing but it's a part of innate immunity.